Hi, thank you very much for tuning in to the OHBM educational course on an introduction to network neuroscience. My name is Janine Bijsterbosch, and I'm going to be talking about defining network nodes, how to represent the brain. So first of all, let's recap and set the stage about what we want to do in network neuroscience. We start off with a really high dimensional complex data set. I'm going to be a little bit focused towards resting state functional MRI data in this talk, but all of the concepts that I'm going to cover are equally important in other modalities as well. The first step that we take in network neuroscience analysis is to define a spatial summary, which is essentially going to take our high dimensional data and summarize it into a lower dimensional set of brain regions, which are going to be our nodes in our graph. The next step is a temporal summary, essentially extracting a single time series for each node in our graph that represents the data for that node. And then once we have that, we have all of the ingredients we need to do our network neuroscience analysis, whether that's looking at all of the connections in the connectome as a whole, looking at changes in the edges dynamically over time, or looking at topological summary features of our graph. The focus of this talk is on the second step, the spatial summary step. And as you can tell, this is really the foundation of all of the subsequent steps in our network neuroscience analysis. So it's really, really important to get the node definition for a network neuroscience analysis right. Let's first of all go over some of the different concepts and terms that are relevant for defining the nodes for our network analysis. First of all, when I'm referring to a node, the goal is to identify a region of the brain that we can think of as a fundamental processing unit. And in the rest of my talk, I'm going to go a lot more over the different considerations for how we can define that. The term parcel is also used, which is essentially the same as a node derived from a brain parcellation. You might have also heard the term region of interest, which is also essentially a node, but often in a different context. Cortical area is a region of cortex distinct from neighboring cortex in function, cytoarchitecture, connectivity, or topographic organization. Interestingly, the term network is kind of confusing and gets used for multiple different things. In a way, a network is an intuitive way of saying a collection of things. And so within neuroimaging and neuroscience, a network might refer to the full connectome, so all nodes and all edges of the brain graph as in network neuroscience. Or in some parts of neuroimaging, we refer to a subset of all of the nodes that are interconnected with each other, so a module as a network. And that kind of relates to the third definition, possible definition of a network within our field, which is essentially a macroscale system of the brain, so a resting state network, such as the default mode network. One of the features you'll see throughout my talk is this section on the right-hand side, where I list a few different relevant papers that you can look at if you want more information. If you download the slides, these links should be clickable, and so it should be easy to find these papers quickly. So here's the outline of the rest of my talk. In section 1, I'm going to go over multiple different considerations to think about when you're defining your nodes. In section 2, I'm going to go more in depth about different methods that have been developed in recent years to define the boundaries of nodes for individual people. And then in section 3, I'll discuss some challenges and caveats in terms of the interpretation in relation to how we define our nodes. So let's start off with section 1. This is an overview of the whole section, and in the next few slides, I'm going to delve into each of these different considerations for node definition in a little bit more detail. The first consideration when thinking about defining nodes is whether you want a hard or a soft parcellation. A hard parcellation, also known as a binary parcellation, essentially assigns either a 0 or a 1 for any single voxel or vertex for any single node. And so that means that a specific voxel or vertex is part of one node and not part of any other nodes. And the advantages of this is that it's intuitive and it's very easy to visualize. But the disadvantages are that it might be a bit of an oversimplification of the functional organization of the brain. And also it might not be a good fit for the noisy low SNR measurements that we get with, particularly with functional MRI, but with other MRI modalities as well. The alternative is to use a soft parcellation, which is also known as a weighted parcellation. And here, instead of assigning a voxel or a vertex as just a 0 or a 1, it can be given a continuous number or a weight. And so that means that a particular voxel or vertex might be able to contribute a little proportion to multiple different nodes. And so the advantages of this approach is that it can capture more complex brain organization, and it allows for measurement error. So essentially, by allowing a little bit of fuzziness in the borders, for example, you can perhaps better capture noisy measurements. The disadvantages of this approach are that it can be a little bit more difficult to interpret and visualize. And in some cases also it can be more difficult to compare across different studies. And that's because the methods used to define soft parcellations are typically performed on the data set itself. A common example is, for example, independent component analysis. And so the breakdown into different weighted networks might be subtly different across different studies, making them a little bit harder to compare. The second parcellation consideration that we're going to talk about is whether nodes should be contiguous or not contiguous. So a contiguous node is essentially a single blob of spatially interconnected voxels or vertices, whereas a non-contiguous node contains multiple different blobs in separate parts of the brain that are not interconnected. The advantage of a contiguous nodes is that it fits most closely with our conceptualization of the brain as a graph, and you can draw lines between different areas in the brain. However, the disadvantages are that it can be inappropriate for some edge-based measures, such as partial correlation. An example of this is if you take the bilateral primary motor cortices, for example. Whenever you calculate using partial correlation the connectivity between the left primary motor cortex and, for example, the supplementary motor area, you would regress out the time series from the right primary motor cortex. And if these regions are super similar to each other and have a very high correlation, which is commonly the case, then you end up removing essentially all of that shared signal, and you might be looking for connectivity in the noise space rather than focusing on interesting signal. On the other hand, advantages of non-contiguous parcellations are that it can capture more complex organizational principles in the brain, for example, bilateral organization or network organization. However, one of the disadvantages are that you lose the ability to study within node connectivity. So for example, if you combine the left and the right motor cortex into a single node, then you can no longer study connectivity between those two regions. The next consideration that we're going to talk about is dimensionality, or how many parcels do we want to cover the brain? And so the question that you might want to ask there is, well, tell me what is the best number of parcels? And the answer to that is that we don't know. We don't have access to the ground truth functional organization that would give us the right answer to validate methods against. And it's useful to remember here that the number of parcels is to some degree limited by biological and physiological limits in terms of the brain and in terms of our measurements. So for example, when we're using resting state function MRI, what we're measuring is a hemodynamically mediated signal. And so the vasculature has an influence on the parcellation that makes sense at that level of measurement. It's also useful to remember that all data-driven solutions are optimized. So essentially whether you use independent component analysis or a clustering approach, all data-driven methods are trying to optimize and find the best possible solution given the constraints of the model. So in a way, all parcellations are appropriate and correct summaries of the brain. The last thing to remember is that there's probably no such thing as a single best dimensionality for the brain, because the brain is organized hierarchical in that you have, systematic organization within individual layers of cortex, but also entire parts of cortex and cortical areas that have the same sensitivities. So there's probably multiple different levels of the hierarchy that accurately represent the brain's inherent organization. So how do we gain insights into this? Well, if you're interested in studying these sorts of questions, I think there's a lot of opportunities for using hierarchical models to link different scales of analysis to each other more explicitly. multimodal research, leveraging different modalities to gain further insights beyond the limits of the measurement of MRI, and potentially testing stability and not homogeneity, because homogeneity within a node is always going to improve as the node gets smaller and smaller, but that's not necessarily what we're aiming for. And so perhaps a better question to ask is, what is the best dimensionality for your specific study that you're about to do? And so for that, I would recommend to try and think about your research question in relation to the decision of the dimensionality. For example, if you're interested in connectivity of the posterior cingulate, you probably don't want that region to be part of the default mode network as a whole as a node, but you probably want it to be its own node so that you can measure its connectivity in itself. Another thing that you can do is test whether your results are stable across multiple different dimensionalities, so that it's not just a fluke result that you only see with one dimensionality, but it's the inferences that you're drawing are somewhat stable across at least a small range of different dimensionalities. And I would advise you not to just use the same parcellation that your lab always uses without thinking about it, because it is the case that for different research questions and different end goals, you might want to use different dimensionalities and different node definitions more broadly. The next consideration that we're going to talk about are the characteristics of the sample of your study. So there are a lot of atlases or parcellations available out there in the literature that you could just pick up and use. And actually, towards the end of my talk, there's a slide that lists a few of them that you can look at. So there is an advantage of using these high quality available atlases in terms of having really good generalizability to other studies that have been performed, and really good replicability if you do a similar study in, for example, a different cohort or different patients. However, the disadvantages to be aware of are that these atlases that are available in the literature are typically based on young, healthy subjects. And so if you're studying children or older people or patients with a particular disorder, then you might want to think about whether the parcellation that you're using is really appropriate and likely to be a good match to the population that you're interested in. And so an alternative option is to use the data that you have that you're going to use for your study to do a study-specific parcellation. So for example, you can use some of the algorithms that have been shared using, for example, either independent component analysis or different clustering approaches that people have developed to actually derive a parcellation that is specific to your study. And so the advantage of that is that you're going to get the best representation of your data. But a potential disadvantage of that is that it can be a little bit harder to relate your results to other papers because the nodes might have shifted a little bit. The last consideration that we're going to talk about is the imaging modality that you're working with. Network neuroscience analysis gets done using resting state fMRI, diffusion data, and other modalities as well. However, the nodes from one modality are not necessarily the best option to use in a different modality. And that's because the borders of structural organization don't always align particularly well with the borders of functional organization in the brain. And so what I would recommend is to use a parcellation that's informed, whether you do it on your own data or whether it's from the literature. Use a parcellation and a node definition that is informed by the modality for your study. It's also a good idea in general to include specific justifications for your choices in this category and in all of the other considerations that we mentioned as well. And one specific piece of advice here is not to use atlases such as the AAL and the Harvard-Oxford Atlas for resting state network neuroscience analysis. And that's because these atlases have been derived on usually quite a small number of subjects based on anatomical landmarks. And it's been shown that those boundaries in those regions don't match particularly well with functional organization and resting state data. And for example, the first paper in the list of relevant papers shows that a little bit in more detail. So in summary, for section 1, I would really encourage you to take the time to think about different node definition considerations, all of the different options that we've listed here and all of their advantages and disadvantages, and make an informed decision for your study. If you rush in and just want to get to the interesting stuff for network neuroscience, you might end up shooting yourself in the foot, because how good the interpretations are really stand or fall with how well designed your nodes are in the first place. It's also really helpful to include a justification of your choices in the paper. As I've shown you, there's almost never a clear right or wrong answer. And so saying what you did and why you did it can really help people understand. And the last thing that you could consider is testing multiple different node definitions. I've mentioned this already in relation to dimensionality, but you could also consider a weighted and a soft parcellation and see how that might change your results and their interpretation. I've talked a lot about how important nodes are for meaningful results from network neuroscience analysis. And one of the things that myself and many others in the field have realized in recent years is that there's a lot of systematic variability in the functional organization from one person to the next. And so that means that if we define our nodes and our parcellation using group averaged or group concatenated information, then those boundaries of that group level parcellation or that group level node might not actually match up very well with the individual level organization. And that's really important, because when we then extract a time series, an average time series within a node, for example, we might be mixing together all sorts of information from different brain regions. And so I want to focus this next section on different methods that have been developed to improve our ability to define nodes that are meaningful, with meaningful boundaries at the individual subject level. This is still a fast-moving field, and there's a lot of new research coming out in this field, but I think it's really important to be aware of how people are thinking about this challenge and what sort of directions people are taking to try and address this problem. The first approach that we can think of for improving our ability to have meaningful boundaries for individual subjects is essentially to improve our registration. So if we do a better job at aligning the brains in the first place, then those group defined boundaries might be more meaningful for each of our people, for each of our subjects. And so on this slide, I'm summarizing two very different approaches to improve registration. The first one is called MSMO, or multimodal surface matching, using all different modalities. And so here the idea is that you can, compared with volumetric registration, you can already get much better registration on the surface. But typically in FreeSurfer, for example, that's only driven by structural information. But actually, if you combine that with resting state information, task activation maps, then you can do a much better job aligning across people those functional differences. And so an advantage of this approach is that it can get really measurable improvements in the sharpness of group average results. So that's shown in the figure on the left here, and it's also shown in the Coulson paper that's listed in on the right part. However, an important challenge with this approach is the known existence of non-diffeomorphic variance across people. So for example, if you have a brain region that's here in one person, but then in another person, it's kind of split up into two regions, and there's another region that kind of goes in the middle of them, then that sort of variation is not something that can ever be captured using smooth warps. The second challenge is that potentially we're losing meaningful information by removing the spatial variability that exists in the first place. So for example, we've shown that those warps that get estimated for MSMO, which are usually just used to transform brain data into a common space and then discarded, those discarded warps actually are really significantly associated with behavior. So it seems that there is some interesting information there that we might be removing. A second and completely different take on the problem of registration is the idea of hyperalignment. And here the method essentially does away with brain space completely, and it doesn't try to align patients in terms of their voxels or vertices. but instead it projects people onto a shared space that isn't the spatial brain space. So you can align subjects by projecting them based on shared activity or brain activation or shared connectivity features. And so the advantage of this is that it gets around these non-diffeomorphic variants because it's no longer trying to align the brains as a whole. However, challenges with this is what information do you use to drive this projection? How do you constrain it? And how do you interpret this? Because we're used to kind of thinking about graphs and kind of making figures that project graphs back onto brains. So another approach that people have taken to be able to investigate more individualized nodes is what I'm calling deep phenotyping. And here the idea is essentially to do away with the group level completely and analyze data at the individual level. And this is done by obtaining hours of data for each individual so that you have sufficient degrees of freedom and sufficient data to be able to robustly estimate a data-driven parcellation just for that individual. And so the advantages of this approach are that it's by far the best way to capture truly individualized nodes. And it really fits with the goal of personalized psychiatry, being able to scan one person and draw interpretable conclusions from just that data set. However, some of the challenges with this is that it kind of lacks correspondence. So often what we end up wanting to do is compare different scans to, for example, compare patients versus control or look at associations with behavior or with symptoms. And for that, you got to be able to compare apples to apples and oranges to oranges. And when you're running a completely separate parcellation for separate individuals, that correspondence of being able to say, is this node in person one the same as this node in person two, can be challenging when you're running these analyses separately. And similarly, generalizing your results to other papers can be challenging, but most importantly, one of the challenges with this approach is high data requirements. It might just not be feasible to get hours of data for because your patient can't tolerate it, or because you can't afford it, or because you don't have the kind of infrastructure to be able to do that. The next category of methods that have been developed to identify individualized node boundaries is to essentially do a post hoc mapping of a group-defined parcellation to kind of redefine each of the nodes based on each individual's data. And there's a number of different methods that fall within this category. Dual regression and back projection are options for independent components analysis defined nodes. But there's also options for machine learning and iterative regression. And so the advantage of this approach is that you make sure by doing the group parcellation in the first place, you make sure that you have correspondence across subjects. And so that makes it easier to compare between different subjects. And another advantage can be that some of these methods can be more easily compared across the literature if you're using the same starting point. However, some of the challenges are still these non-diffeomorphic or non-overlapping variants. If a node is in a specific location at the group level, and it's in a completely separate location in an individual subject, then most of these methods will struggle to find it. And that kind of links to the next challenge with this approach, which might be that some regions might end up being missing in some methods, or actually being made up. So if the post hoc projection is too strongly driven by group level information, then it might find a region regardless of whether it's there or not in that individual. Or if it's too strongly driven by the individual data, you might end up with missing regions, which then becomes really hard to deal with later on within the network neuroscience analysis. The last group of methods that try to get at individualized nodes are integrated models. And so these models are designed to, within the same hierarchical framework, estimate both a group level solution and an individual level solution. And this is typically done within a Bayesian framework, iteratively optimizing both of these aspects and using some group priors to constrain the individual estimates. Advantages of this is that it's a really nice mathematically principled hierarchical model of different types of variability. And of course, we do get that correspondence across subjects because there is some constraint of using the group level information to drive the individual level estimates. However, that's exactly where the challenge is. There's a lot of different parameters in these models, and how do you determine, for example, the appropriate strength of the group prior? You want to make sure that it's strong enough to get that correspondence, but that it's not so strong that we're losing out on finding meaningful individual variation in the nodes. And so to summarize this section on individualized nodes, I would strongly argue that the interpretability and meaningfulness of results from a network neuroscience analysis are much better when we can think of nodes as being meaningful matches to the data at the individual subject level. However, this field of how to best do this is really a moving and developing scientific field. So I'd encourage you to inform yourself about what research has come out recently and what are the options for defining individualized nodes. And although there's no clear best solution, it's probably better to try and do something to get more accurate subject-specific boundaries compared with just using group level boundaries. And lastly, as I had as a takeaway in my last summary, it's really useful if you can include a justification of your chosen method and how you chose it and how you weighed up the different advantages and disadvantages. So in the last section of my talk, I want to discuss a few different gotchas or challenges in relation to interpreting network neuroscience results that come out of these, the definition of the nodes. The first interpretation challenge that I want to talk about is oversplitting. And this is related to the topic of dimensionality that I discussed earlier. And the thing to be aware of here is that our definition of nodes and the dimensionality of our nodes interacts with the method that we use to define our edges to build our graph. So for example, in this figure on the left here, you can see that if region 2 or node 2 is really one homogeneous region, but accidentally for one reason or another, we end up splitting it into region 2A and region 2B, then we end up in when we look at the edges, missing this connection between node 1 and node 2 when we do partial correlation. And that's because when we estimate the connection between node 1 and 2A, we regress out 2B and vice versa. And so that's an example of this node definition leading to false negatives. This is challenging to get exactly right, and therefore one of my recommendations is to, again, include a clear justification of your node definition that matches and is a good fit to your research question, and also avoid over-interpretation of the results and discuss potential caveats. It can also be useful to investigate both full and partial correlation so that you can get a better sense of what information is shared between them and what information is different between those. The second interpretation challenge that I want to talk about is node heterogeneity. And here, when we do network neuroscience analysis, virtually in all possible analysis, we define a single time series for each node. So we represent each node as a single averaged or otherwise regressed time series. However, this is highly likely an oversimplification of the complexity. And so there might be inhomogeneity or heterogeneity within the node. So some recommendations for looking into this further is, first of all, to consider a soft parcellation instead of a hard parcellation, where we can portion out different amounts of variance within the same vertex to contribute to one node and to contribute to another node. Another recommendation or potential option here, if you're interested in this heterogeneity, is to consider doing a gradient analysis to explicitly investigate different kind of modes of organization within a node. And again, I'd encourage you to look at some of these papers on the right here to learn more about this. And the last challenge in terms of interpretation that I want to touch on briefly is representational ambiguity. And here the idea is that when we define our nodes, it essentially determines the lens that we take to look at our data. And to some degree, it limits the types of results that we might get. An important thing to be aware of is that we have shown in multiple different papers that connections or edges between nodes can be influenced by other sources of variability that are not necessarily interpretable in terms of connectivity or coupling. And examples of these are amplitude changes or unmodeled spatial variability. The recommendations to kind of counteract that would be to use, again, individualized nodes, define boundaries of nodes for each individual using that person's data to limit these unmodeled spatial variability influences. And another option that might be really interesting to look into is to use a technique called additive signal change analysis to test for amplitude influences. And that relates to the first paper on the right-hand section. So in summary for this section on interpretation challenges, I'd encourage you to be careful not to overinterpret results from a network neuroscience analysis. There's a lot of caveats in terms of how we decide to partition of the brain that can affect the results that we see. One interesting option is to run additional analyses to test for influences of different types of variants so that you can better understand your results and draw more informative conclusions from your results. And the last recommendation here is to discuss potential sources of ambiguity in the paper, which can be really helpful to further the field and inform a broader discussion about this. So as I said at the start, I have a table here with different resting state atlases. This might not be completely comprehensive, but this covers some of the most commonly used ones. And again, these links to these different papers are clickable if you download these slides. And so if you're looking for using a literature-based atlas or a resting state atlas parcellation, then this is a really good starting point to look at. I've also included a short self-study quiz here. I'm not going to run through these questions, but I would encourage you to look at this because considering some practical examples can be a really good way to learn. And you can find the answers as well as a short explanation on the bottom. Again, there's not always necessarily an obvious right or wrong answer, as you can see throughout my talk, but there are, when you're weighing up the advantages and disadvantages, there's usually a preference for one or the other. And so that's really what this quiz is trying to help you start thinking about. So with that, I want to say a big thank you to the organizers for this educational course, Andrew Zaleski and Alex Fornito. Shout out to my lab, my funding, and my wonderful collaborators. And thank you very much